Hi everyone, welcome back to the channel, Dr. Rasi Explains. This is Dr. Rasi, and today's topic is General Biology 1 Lab 1010L and Module 1. So let's get started. So in Module 1, we have 4 labs. So it start with lab safety, quantification of data, Lab 1, microscope, Lab 2, cells, Lab 3, and chemistry of life, Lab 4. So first, the data quantification and Lab 1. So for all these 4 labs, always check your notes and your records from the lab, all the data that you collected, your lab notebook, and your lab book, which is BSc 1010 lab manual, this orange book. So make sure you read through and review labs 1 to 4 in the book and in your notes all the way to the end of chapter 4, which is page 40. So you should read from page 1 to 40 and review all the stuff. So lab 1 has a lot of introduction stuff and also has data types like binomial or continuous data, and also converting volume, weight, temperature from non-metric system to the metric system, right? So you should be able to do these. So we had data like qualitative data, which was basically something that's actually we don't measure as like as like quantitative data, or versus like eye color, for example, versus quantitative data, like that could be discrete, like binomial, and like you did an experiment like asking people, did you have breakfast? Yes, no. Binomial, making a bar graph, versus continuous data like grade distribution in a bell curve, water temperatures, grades, drill depths, and stuff like that. And also don't forget the specific gravity, which equals to weight of a substance divided by volume of the substance. Okay, so this was basically, and also just in general, the scientific method, that how a scientist actually problem solve. And so they use scientific method, starting with a question, forming an hypothesis, testing that hypothesis with a group of experiments, collecting data, look at the results, come up with a conclusion, and either accept or reject the hypothesis that actually they started with. So this was lab number 1. Lab number 2, which is a microscope, and lab number 2 and lab number 3 go together, basically microscope and cell. Obviously, microscope is the device that we use, which is since 1665. Robert Hooke basically came up with the first microscope. Today we have much complex microscopes like electron microscopes. But in this lab, we use a light microscope, and we can also, do live specimen under the light microscopes. So you need to know all the parts of the microscope. Make sure you check the parts, review each part, total magnification, which is 10 times the objective you're using, what is a bed mount, what is compound microscope, what is electron microscope, and what are objectives. And also you should compare your drawings of a bacteria cell, plant cell, animal cell, and some basic organelles of cell. So we draw potato cell, we draw onion cells, epidermis cells. We also draw Elodia leaf cells. So make sure that you can identify them, and you can also identify organelles and under each of these cells. So that was lab 2 and 3. And lab 4 is chemistry of life, really like food tests. So basically, we have 4 macromolecules to test, including carbohydrates, lipids, proteins, nucleic acids. And under carbohydrates, we have monosaccharide test, polysaccharide test, and we have protein test, we have lipid test, we have nucleic acid test, and we use a specific a solution or a assay to actually test this food to see if each of them has that specific macromolecule that we are searching. So for monosaccharides, we use Benedict solution and look at the color change like the orange. For polysaccharide, we use iodine, and we look at the color change of black. For proteins, we use also a test that was basically which checks actually if we're getting a blue or bluish color from these solutions. For lipids, we have a brown paper that actually we test for their grease accumulation or not. And then we have eventually we have pH and pH scale. You actually did an experiment measuring pH of a number of items. And also always remember in every experiment, you have a negative control, which is usually water, which is usually your very first tube number 1. So, so go over and review and make sure that you pay attention to these color changes, orange or no orange. If you're looking for orange color, any other color is not correct color. So then actually you should, that's your positive result. Or black is your positive result in another experiment. And pH, how we measure the pH using pH paper. So this concludes our review for Module 1 in BSc 1010L. Thank you for watching, and please feel free to comment and subscribe to our channel, Dr. Asi Explains. And please share, and see you in next video. Happy studying.